Okay, where we are now is to discuss what we call the MHC. Where we are now is to discuss the MHC. The MHC stands for Major Histocompatibility Complex. Major Histocompatibility Complex. Now let's break the word down. Histocompatible, tissue compatible, tissue compatible. This is what they check out to see when it comes to transplants, to see if this organ is tissue compatible from one person to the other. All right, now listen to my terminology. The major histocompatibility complex, if I just use this term, major histocompatibility complex, just the major histocompatibility complex, it is the set of genes in your DNA associated with making the protein flags on the cells. I'm going to say this again. So the complex are the genes. The complex are the genes. Now, if we say MHC proteins, that means those are the protein flags on a cell. The protein flags on the cell. Generally glycoproteins. But these are the flags, I'll say, identification flags. So the major identification flags from one person to the other when it comes to transplants is called the MHC proteins. But the MHC proteins were made, cut and snipped off of the blueprint of the MHC, the major histocompatibility complex. So the word complex are the genes and the proteins are what are being made. Now, these are self-antigens, isoantigens, because yours are different than mine, but they would cause an antigenic reaction if put in a human, if put in the wrong person. Transfusions, grafts, these are imparted in that. Okay, so we go further. The MHC proteins are on chromosome 6. There's about 140 genes. There are two MHCs. There's an MHC1 and an MHC2. Let me say this again. There's a major histocompatibility complex 1 that makes MHC1 proteins, and there's an MHC2. For right now, okay, every cell in your human body, listen to me, every cell in your human body has MHC1 proteins except your red blood cells. Every cell in the human body except the red blood cells use the MHC. The red blood cells, remember, used a bunch of different systems, around 31, 32 different systems. And that's where the ABO, RAH, KEL, KIT, Duffy. So they use a separate flagging system. But other cells are using the MHC1. Now, later we're going to find that there are some special cells called antigen-presenting cells that have MHC2. So an antigen-presenting cell, when we get to it, would have MHC1 because all cells do except red blood cells. But additionally, they would have MHC2 proteins that would identify them as some special cells in the immune system called antigen-presenting cells. We won't worry about the MHC3. Okay? All right, we'll go further. I'll ignore this here. So here are the MHC proteins that you have. Okay. An MHC protein, let's say it's like this here on a cell, that'd be the membrane, has a groove in its teeth, has a groove here. So the MHC proteins have a little groove. And in that groove, you put a small peptide in that groove. So this would be the MHC itself. And then in there, we're going to talk about a little piece of protein that's put in there. A little piece of protein that's put in there. Okay. So the MHC surface markers have a groove in their surface. The groove is filled with some substance. if the MHC is filled, if in some cases, now on an MHC1, the one on every body cell, is filled with an endogenous protein. Endogenous means it's made by the cell. So in the groove would be a little piece of protein made by the cell. Please read on this. Now that made by the cell could have been made by the cell through normal action or forced action if it was a virus or tumor. The MHC2, which are only on antigen presenting cells, have something exogenous in their teeth. That's because the MHC2 cells would have done phagocytosis and would have chewed up something to put in the teeth. So it's exogenous. MHC1 endogenous. You got to read on this. I can't explain it all in this video. Okay? But you have MHC1s and MHC2s. MHC1s on every cell in the body except the red blood cells, and MH2 only on antigen presenting cells. Okay. Two types of lymphocytes are in the adaptive system. So really 3 types of cells in this third line. The B lymphocytes, which we'll be talking about, produce antibodies. They're for humoral, extracellular. And the T lymphocytes, cell mediated to take cells out if they're infected by intracellular stuff. Then the third group of cells we just kind of mentioned, antigen presenting cells. So the 3 cells in the third line of defense, adaptive, are B lymphocytes, T lymphocytes, and some cells called the antigen presenting cells. Okay? Lymphocytes originate in your bone marrow. Lymphocytes originate in your bone marrow. Now if you go back to the lymphatic system, we said that you had primary lymphoid organs and secondary. The primary were where lymphocytes gained education, immunocompetence. So all lymphocytes are made in the bone marrow. But where do they gain their education? If they gain their education, i.e. immunocompetence in the bone marrow, they become B lymphocytes. If they leave the bone marrow, do not get educated in the bone marrow, then they may pass through the thymus gland and get educated. If they get educated, immunocompetence, i.e. in the thymus gland, they're called T lymphocytes. So B lymphocytes were educated in the bone marrow, T lymphocytes were educated in the thymus gland. B lymphocytes are the ones that produce antibodies, and T lymphocytes do cell-mediated immunity. Okay? If they don't get educated either place, they may get killed off, or they may become the second line lymphocyte known as a natural killer cell. So we look here natural killer, T. Now there are different kinds of T cells we'll talk about. Different types we'll talk about. So let's say natural killer, T lymphocytes, and B. So you can see by far T lymphocytes predominate in your body. Okay? So, when in the education process of T lymphocytes, now okay, so let's go back. All lymphocytes were made in the bone marrow. Let's say they don't get educated in the bone marrow. So then they leave and they go through the thymus gland and they get accepted to the thymus gland school. Then there are 2 major courses. Let's say this is year 1 and this is year 2. It doesn't take that long. I'm just joking. But 2 major things they must pass. The course on immunocompetence and the course on self-tolerance. The course on immunocompetence and the course on self-tolerance. Immunocompetence basically means they must be able to recognize how to look at a cell and determine which flags to look at to say if it's part of them or part of something else. That's immunocompetence. They're able to recognize and bind to specific substances on the cell very loosely. So the immunocompetence is to know what to look at, is to educate the cells to know what to look at. These potential graduate T lymphocytes. Self-tolerance is do not attack yourself. Do not attack yourself. So here's the terminology that you'll see. Before immunocompetence, that means you're coming straight out of the bone marrow and you have not been educated. So before immunocompetence, no education, the cells are immature. The newly released lymphocytes are immature. After immunocompetence, but have not met an antigen. So you've gotten educated, but you haven't seen anything to fight. Then you're naive. After meeting an antigen, but no co-stimulation, we'll talk about that. So in other words, when you meet an antigen that you recognize, you still can't fight it unless you get confirmation from another cell. So you would have the antigen, but you can't fight it. You can hold it, but you can't fight it. So I'm going to call it active idling. And then after co-stimulation, fully active. So, but for T cell, that means it comes out of the bone marrow, let's say, immature. Now it goes through the education process. But T cells, there are two areas of education: immunocompetence, first course, and then self-tolerance, we'll talk about. The primary lymphoid organs are the thymus gland and the bone marrow. The primary lymphoid are where it gets educated. Remember, secondary are where you go to after it's become educated to populate the thymus gland, tonsils, stuff like that. So this is a nice little drawing to show you leaving the bone marrow, being educated in the bone marrow, and then going to work in some of these secondary, or being educated in the thymus gland. You can read on that. Okay. T cells, T lymphocytes, have a T cell receptor. The T cell receptor is a CD3. So if a cell has a CD3, it's a T cell. If a cell has a CD3, it's a T cell. Clusters of differentiation. CD3 is a T cell. So when a T cell, or when a cell from the bone marrow enters the thymus gland, it undergoes the education process. Under the education process, two things you want to happen: positive selection and negative selection. Positive selection and negative selection. So we're going to start right here with the T cell education again in our next video.